Hi everyone, welcome to today's lecture. In the beginning of this chapter, we learned about two types of diseases, that is infectious diseases and non-infectious diseases. Infectious diseases is the diseases which is transmitted from one person to the another. The example for this is AIDS. And the non-infectious diseases are the diseases which are not easily transmitted from one person to the another. The example is cancer. Today we shall discuss about these two diseases in detail. First, we shall discuss about AIDS. The word AIDS stands for acquired immunodeficiency syndrome. This means there is a deficiency in the immune system which is acquired during the lifetime of an individual. The word syndrome means a group of symptoms. The AIDS was first reported in the year 1981. It has spread to all over the world, killing more than 25 million people. The AIDS is caused by a HIV virus. The HIV stands for Human Immunodeficiency Virus. This is a member of a group of viruses called retrovirus. This virus contains RNA as a genetic material and this is an enveloped virus. Now we shall talk about the transmission of HIV. This infection generally occurs by sexual contact with the infected person, by the transfusion of contaminated blood and blood products, is by sharing infected needles, as in the case of intravenous drug abusers. Fourth one is from the infected mother to the child through the placenta. But HIV or AIDS are not spread by physical contacts. It only spreads through the body fluids. The incubation period may vary from few months to many years, usually 5 to 10 years. Next, we shall understand how retrovirus replicates. The virus enters the body of a human and reaches to the macrophages. Macrophages is one of the cells of our immune system. The viral RNA is converted to viral DNA with the help of an enzyme, reverse transcriptase. Then the viral DNA incorporates into the host cell's DNA and directs the infected cells to produce virus particles. The macrophages continues to produce viruses. In this way, acts like a HIV factory. Simultaneously, HIV enters into helper T lymphocytes, replicates and produce progeny viruses. The progeny viruses are released in the blood, attack the other helper T lymphocytes. This is repeated, leading to a progressive decrease in the number of helper T lymphocytes in the blood of the infected person. Since the number of helper T lymphocytes decreases in the body, the person becomes susceptible to infections of bacteria, especially mycobacterium, and viruses, fungi, and even parasites like toxoplasma. The patients get immunodeficient and he is unable to protect himself against these infections. This is what I explained. Now we shall know about the diagnosis and treatment. A widely used diagnostic test for AIDS is ELISA, enzyme-linked immunosorbent assay. The treatment of AIDS with an anti-retroviral drug is only partially effective, which can only prolong the life of the patient but cannot prevent death, which is inevitable. Lastly, we will move on to the precautions. As AIDS cannot be cured, the prevention is the only best option. In our country, National AIDS Control Organization (NACO) and other non-governmental organization NGOs are doing a lot to educate people about AIDS. The WHO has started a number of programs to prevent the spreading of HIV infection. There are few steps that are taken to prevent these diseases. They are making the blood from blood banks safe from HIV. The second one is the ensuring the use of only disposable needles and syringes in public and private hospitals and clinics, controlling drug abuse, and finally promoting regular checkups for HIV in susceptible populations. This is all about AIDS. Now let's talk about cancer. Cancer is a genetic disorder which are not inherited from parents to offspring. They are called as genetic disorder because of the presence of a mutation in the sequence of DNA. Cancer is one of the most dreadful diseases of the human beings and is a major cause of death all over the world. More than 1 million people suffer from this disease. Cancer is a disease which is caused by the uncontrolled growth of abnormal cells and it spreads to the other parts of the body. In our body, cell growth and differentiation is highly controlled and regulated. But in case of cancer cells, there is a breakdown of these regulatory mechanisms. The normal cells show contact inhibition, thus inhibits the uncontrolled growth. Contact inhibition is a biological phenomenon where the cell growth is stopped when cell makes a physical contact with one another. This regulation helps maintain a monolayer of cells, preventing uncontrolled growth. But the cancer cells lack this ability. As a result, cancerous cells just continue to divide, giving rise to masses of cells called tumors. Tumors are of two types: benign tumor and malignant tumor. Benign tumors normally remain confined to a particular location and they do not spread to other parts of a body, and this causes a little damage. On the other hand, malignant tumors are a mass of proliferating cells called neoplastic or tumor cells. These cells grow very rapidly, invading and damaging the surrounding normal tissues. As these cells actively divide and grow, they also starve the normal cells by competing for vital nutrients. When the cells from the tumors get clogged, they reach to the distant sites through the blood, and wherever they get lodged in the body, they start a formation of new tumor. This property is called as metastasis. We shall talk about the causes of cancer. Transformation of normal cells into a cancerous cells may be induced by physical, chemical, or biological agents. These agents are called as carcinogens. The physical agents are the radiations such as ionizing radiation like X-rays and gamma rays, and non-ionizing radiations like UV causes DNA damage leading to a neoplastic transformation. And the chemical carcinogens are present in the tobacco smoke, which are identified as a major cause for the lung cancer. And the biological agents are the viruses. Cancer causing viruses called oncogenic virus have genes called viral oncogenes. Furthermore, several genes called cellular oncogenes, c-onc or proto-oncogenes, have been identified in the normal cells, which gets activated only under certain conditions, which leads to an oncogenic transformation of the cells. Next, moving on to the detection of cancer. The early detection of cancer is very essential as it allows the disease to be treated successfully in many cases. Cancer detection is based on biopsy and histopathological studies of tissues and blood, and bone marrow test is for increased cell count in the case of leukemias. Histopathological studies means it is the microscopic examination of the diseased tissues to understand and diagnose the conditions. In biopsy, a piece of suspected tissue is cut into thin sections and it is stained and examined under microscope by a pathologist. Techniques like radiography, use of X-rays, CT and MRI are very useful to detect cancers of the internal organs. Computed tomography CT uses X-rays to generate a three-dimensional image of the internals of an object. In case of MRI, the magnetic resonance imaging uses strong magnetic field and non-ionizing radiations to accurately detect pathological and physiological changes in the living tissues. Antibodies against cancer-specific antigens are also used for the detection of certain cancers. Techniques of molecular biology can be applied to detect genes in individuals with inherited susceptibility to certain cancers. Identification of such genes may be very helpful in the prevention of cancer. Such individuals may be advised to avoid exposure to the particular carcinogens to which they are susceptible. For example, tobacco smoke in case of lung cancer. Lastly, we shall discuss about treatment of cancer. The common approach for the treatment of cancer are surgery, radiation therapy and immunotherapy. In radiotherapy, tumor cells are irradiated lethally, taking a proper care of the normal tissues surrounding the tumor mass. Several chemotherapeutic drugs are used to kill cancerous cells. Some of these are specific for particular tumors. Majority of drugs have side effects like hair loss, anemia, etc. Most cancers are treated by the combination of surgery, radiotherapy and chemotherapy. Remember, immunotherapy and chemotherapy are not the same. While both are cancer treatment, they work through different mechanisms. Chemotherapy uses drugs directly to kill fast-growing cancer cells, while immunotherapy utilizes the body's immune system to recognize and attack cancer cells. Tumor cells have been shown to avoid detection and destruction by the immune system. Therefore, the patients are given substances called biological response modifier such as alpha interferon, which activates their immune systems and helps in destroying the tumor. Therefore, the patients are given substances called biological response modifier such as alpha interferon, which activates the immune system and helps in the destruction of the tumor. In the next lecture, we shall discuss about drugs and alcohol abuse. Thank you.